Okay, so slide number 6 finished. Now we go through the slide number 7. We should start from this point as you tell me. Positive interactions. We want to have some examples. Now we want to have some examples when we have only one population. For example, suppose there is one flask or there is one tube. Inside the flask or tube, only we have E. coli. All the members are E. coli. All are the same. There is no any difference between them. So when food is sufficient, there is positive interaction or cooperation. We want to have some examples. In page number 7. First, we talk about lag period. Lag period. So what is this? Suppose there is a bottle of milk. You put one E. coli in the milk. What do you think? So immediately one E. coli doesn't start eating. Immediately doesn't start growing. Immediately doesn't start multiplication. First, only look the environment. Which substrates are present? Which enzymes needed to be expressed? For example, suppose in one environment only glucose is available. It is not wisely the bacteria produce lactase, sucrase, maltase, cellulase, and many enzymes which substrate is not available. Okay? So this part is lag period. In lag period, they feel the environment. How do they feel? They have some receptors on the cytoplasmic membrane. And if there is lactose, so receptors of lactose occupied, signal going to the DNA, DNA order the ribosome to produce lactase. If sucrose is available, sucrose attached to its special receptors on cytoplasmic membrane, signal going to the DNA, and DNA ordered ribosome produce sucrase. So it takes time. So the time which a microbe coming to a new environment and don't do anything, only look around, only feel what is present and what is not present. According to the environment, some genes will be on and will be expressed, and some genes will be off and not to be expressed, not working. So you accept from me that there is a lag period. Sometimes 10 minutes, 15 minutes, something takes time. As much as the microbes come, the same microbes come to the new environment, lag phase is shorter. And as much as the number of the same microbes coming to the new environments are less, lag phase is longer. And if very few number of the same microbe or one microbe come to new environment, lag period not to be finished, and microbe cannot go to the logarithmic phase. It's remaining there. So what can we understand according to this sentences? Again, repeating. If the number of inoculants, the group coming to the new environment, we say inoculant, are less, lag phase is longer. And in opposite, as much as the number of population coming to the new environment are more, lag phase is shorter. So it is good. This sentence explain when there is population, as much as the number is more, they can start using the substrate, can multiply, can going to the logarithmic phase. Okay? So, as much as the number is more, they help each other to start absorbing food, to start going logarithmic phase. An extended lag period or a complete failure of growth when a very small inoculation is used in routine culture, mostly about fastidious microorganisms. What can you understand? If the number is low, there is no growth. If the number is high, so there is growth. So we understand when we have population, when we have group, they can help each other and they can grow. So it is positive interaction. So it is cooperation. Is it clear? Can I go to the next? Did you get the point? Yes? Okay. Did you remember in one time when you have requests, so you are 30 students, for example, 30 messages, similar messages, writing and writing and writing. What do we feel? You feel that when in group you write, your power is more than one single person I am telling according to the to my friend's idea. Yes. So in group, when in group you say the idea, it is more strongly. Yes. So when microbes in group coming to new environment can start life, but if their numbers are very low, they cannot grow. For example, if you inoculate one E. coli in milk, no growth. But sometimes you think one E. coli in one bottle of milk, why not growing? Not growing. So the numbers should come to suitable number of population. Population can grow. One single cannot grow. Next example, minimum infectious dose. So those students which has advanced microbiology last 1 hour ago, we explained minimum infectious dose. What is this? The minimum pathogenic microbes which should come to my body to infection, to continue infection, and make a disease. If the number is low, There is no disease, and immune system suppress them. So what can we understand? If single, for example, if one Salmonella typhi come to my intestine or one Vibrio cholera with infected water, I drink, I don't get disease. I don't get disease with many low number of Brucella abortus or Vibrio cholera. But if their numbers are high, they can occupy my intestine and invade. So what can we understand? When the number of a population is low, they are weak. But if their numbers are more, they are stronger. Stronger, weaker. Stronger, weaker. Low population, weaker. Higher population, stronger. So what can we understand? Where as much as the number of population are more, we can see better the positive interactions. We can see better the cooperation. When the number of pathogen is low, our immune system very easily suppress them. When the number of pathogens are high, those pathogens can suppress our immune system. Minimum infectious dose of pathogenic microbial populations. A single pathogen rarely able to overcome host defenses. Okay? But in group, that pathogens in group can overcome host defenses. What can we understand? We can understand there are positive interactions in group. Note, in first example, I explained that if you put one E. coli in milk, not growing, now we want to see why. Why not? Why, if we have one bacterium in milk, why cannot grow? Cooperation occurs within the population because the semi-permeable cell membrane of microorganisms are imperfect. So we know that cytoplasmic membrane is semi-permeable, but it is not working perfectly and tend to leak low molecular weight metabolic intermediate. So many good materials leak outside the cell. That they are essential for biosynthesis and growth. They are essential for biosynthesis and growth, but they are leaking. So when we have only one E. coli, many leaks happening and many good things going outside the cell, and cell feeling hungry and cannot continue the life. But when we have a group, if bacterium number 1 has leakage and lose some material by leaking, the others absorb it. So number 1 leak, number 2 absorb it. Number 2 leak, number 1 absorb it. So if there are leakage in population, but mostly again reabsorbed, but mostly again can be reabsorbed. Again can be reabsorbed. But when the population are low, most leaked low molecular weight metabolic intermediates go far from the bacterium and cannot be reused. Okay? So it is another thing. Excuse me, only 1 second. Okay, excuse me. Continue. Another example. When you inoculate one sample on blood agar, on nutrient agar, on murerinton agar, on any medium, you have many colony. So bacteria like to have colony, even though some of your bacteria have flagella and can have motility. Okay? But they form colony. Colony formation is an adaptation based on cooperative interaction with a population. So all of you have motorcycle or car or vehicle, or all of us, we have feet. We can go far from the population, but why do we live in city? Why do we live in village? Why don't we go far from the other? Because there are many benefits to be in a population. You have the population in your country, you have the population in your city, you have the population in your home. You like to be in population because population give you the power. Population have many benefits for you. So bacteria on plate can go far from each other, but they don't do that. Even though they have flagella, they make colony together. They have population like city, like village. They are with each other because there are cooperative interactions in colony. Even motile bacteria that could move away often remain in colonies. So it is another example of positive interactions or cooperation in one population. Microcolonies rather than individual microorganisms are normally adhering to particles. Even though in any flask, in any tube, which you have nutrient broth or water or what else, many microbes are single living in the broth, but some of them going and attach to the glass, inside glass, and make microcolony. Make microcolonies. They like to be together, to live with population. Suppose when one person has surgery and one person is in the bed in the hospital, there is necessary to use urinary catheter. So the person by serum injection has receiving many antibiotics, and these antibiotics are coming to the urine, and urine by urinary catheter going to the plastic bag, because the person cannot going and come back. All the times, day, night, the person is in the bed, in the hospital. Okay, by urinary catheter, urine going to the bag. So many microbes in the urine can be present that antibiotic kill them. So most of them like to attach to the inside plastic wall to make colony, because colony and group are more resistant to antibiotics than they are single in the liquid. So they produce microcolonies. Okay? So, and sometimes some people has kidney failure, kidney problem, and they must have dialysis. During dialysis, it is a device for purification of blood. Sometimes this instrument is infected by some bacteria, and some of them attach to the wall of instrument by microcolonies, because colonies on the surface are more resistant to the antibiotics and adverse effects. So again, telling microcolonies or colonies on solid surface or liquid surface give advantage, give power. To have microcolony or colony is positive interaction in the population. It is cooperation. And sometimes when we have colonies and bacteria wants to have motion or movement, they have mass movement. For example, Proteus is a bacteria which they have mass movement. We call them swarming, group movement. They go with each other and stop with each other. Again going, again stopping. Again going, again stopping. Like army. They go and stop with each other. Not single. Group movement. Group movement. So it shows to be in a group in colony, to going in group has positive interactions. There is a cooperation. It is beneficial. They like it. Another example is related to the mold. There is a mold with the name of slime mold of Dictyostelium. Slime mold of Dictyostelium. So when there is a food limitation, this mold changing to the amoeboid cells. Some things look like amoeba, okay? And come together. So pay attention. When there is not sufficient food, these cells come together. So they think if they come together, it is positive. They come together for cooperation. When they come together and they come around a central organism by production of cyclic AMP, they can form a special body with the name of fruiting body. And this fruiting body is a special part used for spore formation and disperse them. So this fruiting body produces spore, and the spores going far and in suitable place can change to mold shape. So when they feel danger, they come together. Why? Because when they can come together, now they can produce a spore to escape from death, not to be killed. They change to a resistant form of spore. Another example regarding positive interactions or cooperation. Colonies are important for utilizing insoluble substances like lignin, cellulose by production extracellular enzymes. See, for example, one door is locked and you don't have key and you should open it, but alone you cannot break the door and go inside because it's important to open the door. But if 5 people coming by each other can break the door and go inside, so one person cannot. Now think there is a polymeric food outside bacterium. So bacterium doesn't have any mouth like human. They don't have mouth for eating. They must send extracellular enzyme outside, make the polymer smaller, as much as small can come inside bacteria by passive diffusion, by active transport, by many types of transfer. So they must send extracellular enzyme. So they have 2 types of digestion: outside digestion and inside digestion. First, polymer outside the cell should be cut to smaller parts. For example, if you have a big pizza in front of you, first you should cut it. Then you put a small piece in your mouth, again cut it. Then very small parts come to your intestine. In bacteria is the same. First, polymer outside should be cut by extracellular enzyme. But if there is only one bacterium, very small amount of exoenzyme is not sufficient to produce the needed amount of substrate. For example, cellulose is outside bacterium, and bacterium has cellulase, but very low amount of cellulase cannot produce glucose, sufficient glucose for life. So one bacterium will die. But if there are many hundred bacterium coming together to cellulose, and they produce cellulase, by outside digestion of cellulose, sufficient glucose can be achieved. So now you understand, if there is lignin, cellulose, or there is milk, why one bacterium cannot use it? Because a group needed to produce sufficient exoenzymes. By this exoenzyme, sufficient glucose produced, and they can use it and continue the life. Okay? So 1 microbe, 2 microbe, 5 microbe is not sufficient. They should have a group with positive interaction, have a cooperation, help each other to live there. Colonies are important for utilizing insoluble substrates, example lignin and cellulose, by production extracellular enzymes, but with low population densities, these products liberated. Example, growth of Myxobacterium xanthus on insoluble casein. If we have this bacterium and inoculate in a medium containing insoluble casein, there is no growth by densities lower than 1,000 per milliliter. So when do we have growth? If the number of Myxobacterium xanthus is higher than 1,000 bacteria per milliliter. So they can grow. What can we understand? If they are more than 1,000 per milliliter, by positive interactions, by cooperation, they can use casein and receive sufficient substrate for life. If they are less than 1,000, it is not so, and all of them will die. But if we hydrolyze casein chemically, so there is no need for hydrolyzing. So less number can use pre-hydrolyzed casein. Is it clear? Did you understand why one bacterium cannot continue life in milk, but more than 1,000 microbe can continue life? Because when they are grouped, there are sufficient enzyme, exoenzyme. With sufficient exoenzyme, there are sufficient glucose. With sufficient glucose, they can continue life. And in opposite direction, when they are less, for example, 1 E. coli or 1 Myxobacterium, very low amount of exoenzyme, very low amount of substrate, and it is not sufficient for life. Is it clear? Can I go to the next page? Do you have any question from this page, number 7? Okay, thank you. Page number 8. A few examples, we continue, and then we go to the next part. Sometimes food is there, but it is insoluble. For example, sometimes insoluble phosphate is there, but microbe needs soluble substrate. Some bacteria can produce organic acid and solubilized inorganic elements and use them. But solubilization need group. Solubilization in group is sufficient. If 1, 2, 5, 10 microbe going and try to solubilize element cannot do that, like previous example. Another example is protective mechanism. Protective mechanism. A given concentration of metabolic inhibitor has less effect on a dense cell suspension than on a dilute one. As much as the number of a population is more, the time needed for killing them is also more. As much as the number of one population is more, they are more resistant because they have protective mechanism, because there is a positive interaction between them, because there is a cooperation between them, and when they are in group, they are more protected than they are single. Population within biofilms, I explained you, for example, when one person is in the hospital, need to connect urinary catheter because cannot go outside bed for urination. So urine sent to the bag connected to the urinary catheter. So I explained that inside urinary catheter, some bacteria attach to the inside surface of the catheter and make colony. We call these colonies as biofilm. We call these colonies inside the tube as the biofilm. So biofilm is a group are more resistant to the antibiotics present in the urine than those which are single in the liquid. High densities of populations shield some members from UV. For example, suppose you have two types of colony. One bacterium is older and it is a big colonies. One is young colony and is flat. And then you open the lid of the Petri plate and turn on the UV. Which one easily can be killed? So the big colony is more resistant to the UV because the upper bacteria shield, not allowing UV going to the inner part of the colony. Shield it. But the flat colony, most bacteria are exposed by the UV and mostly they are killed. So as much as the colony is bigger, is more protected and have a more protection activity regarding to the effect of UV radiation. The pressing freezing point of water. So if there are very single number of microbe in the water, easily by freezing, they will be killed. But when they are group, as much as the group are more and more, their resistance to the freezing point is more. Freezing points like North Pole, you can see, for example, penguin come together very close to each other and stand in a very cold, but if they are single, all of them will be died. Yes, but they come together very close to each other, stand in the cold condition. Bacteria are the same. So as much as the group of bacteria are more, they suffer the freezing point easier. And the last example of this part is genetic exchange. Genetic exchange. Genetic exchange is a good thing for bacteria because bacteria understand it very well during many millions of years that they have two types of resistance. One type of resistance is personal resistance, and the second one is group resistance. Okay? So they understand when one is powerful and friends are powerful, so it is cooperation, good cooperation, positive interaction. They can have a good power for continued life. Okay? And they understand if one bacterium is powerful and some bacteria are weak, they should send their powers to them, remaining powerful, and tell the others how they can be powerful also. How do they do that? By genetic transfer. By genetic transfer, by genetic exchange. Genetic exchange can be done by transformation, by transduction, by conjugation. 100%, I think you know in microbiology 1 and 2 as you passed. So if one bacterium has the power of resistance to one antibiotics or many antibiotics, they send this power to its friend which they don't have it. Some bacteria are resistant to heavy metals. Some bacteria have ability to utilize unusual organic substances. So if one bacterium has one or some of these powers, can send this power to friends by genetic exchange. By genetic exchange. By transformation, transduction, conjugation. So when transformation and conjugation is done better, as much as the population is more. How one bacterium can give the power of resistance to friend when the population is more and more. As much as the population is more, sending power is easier. And if the number is very less, if the number is less, there is no possibility for genetic exchange. Example, resistance to antibiotics, heavy metals, ability to utilize unusual organic substances can be sent by genetic exchange. Genetic exchange occurs in high population densities. So genetic exchange is a very good phenomenon for bacteria. When it can be happened, when there is a high population density, more than 10 in power of 5 per milliliter of the bacteria. So it is another example. And during conjugation, for example, one bacterium has the antibiotic resistance, we call it F+. And one bacterium doesn't have this plasmid, what do we call it? We call it F-. So what happened? What happened? F minus is recipient cell. Recipient cell. Don't forget, we are talking about a population with same individuals, with same members, with same strains. For example, recipient cells of Enterococcus faecalis produce a chemical with the name of pheromones. Pheromone means this is a chemical for requesting. So F plus should understand who requests, who need, who wants. So those bacteria without this plasmid send message. Send message by pheromone. It is a chemical substance that induce plasmid bearing donor cells. Plasmid bearing donor cells means F plus to synthesize agglutinins. So F plus synthesize agglutinins, proteins, and sex pilus, and thus to form mating aggregates. Then F plus go and find pheromone producer, attach it, and then send one copy of plasmid to the recipient cell. Where it happened? It happened in population. If you have only 2 bacteria in one tube, never don't go to each other for genetic exchange. At least 10 in power of 5 bacteria in milliliter should be present in tube or flask for genetic exchange. So genetic exchange is a positive phenomenon. This positive interaction happened when we have population, and a population greater than 10 in power of 5 per milliliter. Okay, so all of these things in page 7 and page 8 till this point explain that in population, when there is sufficient food, there are positive interactions and there is cooperation, and all members of the population help each other to live better on that habitat. But sometimes food is not sufficient, so the interaction changed to new negative interaction that we call it competition for nutrients. And on the other hand, after a period of time, toxic substances produced during metabolism and excreted out by one bacterium outside in the habitat is toxic to all population, to whole population. So 2 problems happens for bacteria after some time. 2 problems happening for one population after some time. After using food, and when there is not sufficient food, they go through competition. It is one problem for bacteria. The other one, if one thing is toxic substance after metabolism for one microbe, it is toxic for all, because we have one population and all microbes are the same. If one matter is good for one, is good for all. If one matter is toxic for one, is toxic for all. Okay? So these two things. First, we go through competition. First, we discuss regarding competition. Negative interactions in a microbial population are called competition. All members of one population utilize same substrate and occupied same ecological niche. So when one microorganism uses one metabolite, then it won't be available for others. In low concentration of substrate, increased population cause competition. For example, example 1. Suppose we have predators like Paramecium. We have one tube containing, for example, Paramecium, and the food is bacteria. When bacteria is not sufficient, so all Paramecium go through competition for food. Or parasitic microorganisms. When there are many parasites in or on one host and there is not sufficient food, they go through competition. They go through competition for food. So it is very easily can be understood which competition is negative. Okay, we are talking when we have one population and it is not sufficient food. Do we feel well? Do they feel well, or do they feel negative, or do they feel neutral? Suppose in shops one food is not sufficient. What do people feel? Feel well or feel bad? Or feel nothing? Feel bad, yes? Suppose I have one child and in the city I cannot find the powdered milk, Cerelac, Biomeal, or another things. So what do I feel? I feel negative. Yes? So when food is not sufficient, all members in the population feel negative. Suppose there is 2 child or 10 child, there is one ice cream. One child, there is 10 children. One child take the ice cream and running, and 9 children in behind. Who feel well? No one. The one which have the ice cream all the time feel the other come and get it. And the others feel the first one, it's the ice cream. All feel bad. Suppose in the running, the first child eats the ice cream. Then what? Okay, stop. And the others stop. Those didn't receive it, don't receive ice cream and lose many energy. And the one which eat ice cream lose many energy. If there was one ice cream and one child, it go and sit behind the table and eat it. So it is good, positive. But when you escape, you lose many energy. The one eats the food, it is negative relationship. The others cannot eat it, it is negative. So in competition, never, we don't think One is positive and the others are negative. All are negative in relationship. Okay? We don't talk who eat and who not eating. We tell what is the relationship of the population. The relationship of the population in competition is negative. Negative for all. Even those eating, even those not eating. All feeling negative. Okay? So it was regarding competition. Toxic substances is very easy. During metabolism, when one microbe produce some substances toxic, yes? When it is toxic, excreted outside, but outside is beside the others, and the others excreted outside beside this one. So, toxic substances of metabolism, okay, have negative feedback. Negative feedback. For example, suppose you have a tube or flask. As much as bacteria need food, give them. Okay? But after some time, you can see they cannot grow. Why? Because growth need two wings. One wing is eating, second wing is removing toxic substances. If there is food in the tube, as well as toxic substances, there is growth. All to be killed. All will be killed. So now we can easily understand why Earth is not full of bacteria. Because microbes and bacteria have negative feedback on themselves by their toxic substances coming from their metabolism. Okay? Suppose theoretically, if we give as much as one bacterium need food, we have one bacterium, we give it as much as food needed, and we remove all the toxic substances. After 2-3 days, we have a mass 4 times bigger than the Earth. So why bacteria cannot grow continuously? Because any population of microbes have negative feedback on themselves according their toxic substances. Okay? Clear? Another negative interaction is toxic production by members of one population. It may be accumulated to inhibitory level even when substrate is still available. Some example, for example, sulfate reducers during reduction of sulfate produce a toxic substance with the name of H2S, and H2S stop the growth by negative feedback. Lactobacillus during the growth produce lactic acid, and after some time lactic acid has negative feedback on lactobacillus. Saccharomyces produce ethanol, and after some time ethanol suppress the growth of Saccharomyces by negative feedback. Hydrocarbon biodegrading bacteria produce fatty acids, and fatty aniline has negative feedback on penicillium, Piscarium. Yes? So any populations are the same. If you want to have one population with logarithmic phase, with continuously growth, you should think about two things. First thing is how you can give as much as they need food. The second, you should think how can you remove the toxic substances which they produce after metabolism. It is the same for human. For example, when we eat food, we drink milk, we drink water, we have feces, we have stool, we have urine. So we try to make them far from ours by many tools. We send them far from ours, because if we cannot do that, we cannot continue the life. In our city, we have some systems for sewage runoff to the outside the city. Yes, human disposal going outside the city, so we can continue the life. Food is one thing, and to get rid of the sewage and those things is the another thing. And the last part, which we explain and we finish this lecture in negative and write Escherichia coli Nobel Prize. And then you understand how much this bacterium is helpful for experimental research. So this bacterium was used for research, and the scientists understood that two different genes are present in this bacterium with the name of Hoke gene and Soak gene. Hoke is the abbreviation of host killing gene. Host killing gene. And Soak gene is the abbreviation for suppression of killing. So in normal situation, this gene is active. Soak gene. In all bacteria, especially in Escherichia coli, Soak gene is active and is expressed and produce proteins that blocks Hoke gene. So this gene is not working in normal situation. Okay? When the food is not sufficient and competition is high, in high competitive reactions, some bacteria lose the plasmids containing Soak gene. So some bacteria, because plasmid is very important, there are many resistances are related to the plasmids. Resistance to the antibiotics, resistance to the heavy metals, and so and so. So some bacteria find that they lose their plasmid and they are weaker in the population. So when it happened, this gene is activated. This gene is host killing gene. Okay? And when Host gene is activated, it is expressed and some peptide is produced, which this peptide attack to the cytoplasmic membrane of Escherichia coli and with great damage to transmembrane potential leads to the death. So again, we repeat it. Suppose there is not sufficient food. So 100%, some bacteria will die because, for example, we have 1,000 bacteria and 100 food. So 100%, most of them will be killed. During the pressure of food limitation, some bacteria lose their plasmids which contains soak gene. Without soak gene, Hook gene will activate and express and order for self-killing. So if it is needed, or if 100% in a short time, many of bacteria will die, some of them will be volunteer. Some of them which they are weaker because personal existence and population existence they understand. When there is a danger to the population, some weaker ones more about than person, that personal life according to the belief of bacteria. Okay, we don't talk about human. Bacteria think that the existence of population is more precise than existence weak, and stronger ones don't kill weaker ones. Weaker bacteria going out the population for existence of the population. These plasmids also contain genes for antibiotic resistance, so those defective cells are killed by themselves. The weaker ones kill themselves. The stronger ones don't attack them. The stronger ones don't tell anything to them. The population density is limited because some cells, those that lose plasmid, with hope and so kill themselves. So the food can reach to the stronger one and the population can be continued. Is it clear? Or is it any question? Did you get the point? Okay. So it is sufficient for today. Next time we go through the interactions between two different populations. Today only we think about interactions inside one population. All the members were the same. But next time we have two different populations. We have different population. In today we have only cooperation and competition. But next week we have neutralism, commensalism, synergism, mutualism, competition, amensalism, predation, parasitism. So the interaction between two different populations are more complex, are different things which we want to discuss next week. So please tell me, we are in page 10. Please next week remind me to start from this page. Thank you very much. So hopefully we continue our lectures and we try to finish on time, and hopefully this semester you finish very easily. Third semester, and next week hopefully you start your thesis and get your certificate and you receive the benefit of certificate. So it is finished. Thank you very much for your presence. I'm happy to be with you again. Thank you. Goodbye. See you.